Okay, great. So, hmm. Can everyone see this? Yeah, I apologize for any stumbles or stutters that I might have, but as Will mentioned in the last talk, it is difficult to do this from home. You miss the energy of the crowd, you miss the perplexed or nodding faces, so it's a little bit hard to tell if what you're saying makes any sense to everyone. So I apologize in advance. My lab, so my name is Jason Rihel, and I'm at UCL, and I study sleep in zebrafish. And I've been doing that pretty much for a very long time now. So in today's outline, I'm going to try to tell you about two stories. This depends a lot on how well and smoothly my talk goes, but the first, I'm going to tell you about how we measure sleep homeostasis in zebrafish larvae and trying to get at the mechanisms by which sleep homeostasis works. And the second will be to describe some new computational methods for bridging short and long time scale behavioral measurements in larval zebrafish in a high throughput manner. So let's start with some sleep. We all sleep. In fact, survey after survey, I've taken one here from 2013 when the average American day shows that we spend more time sleeping than we do anything else. No matter what, it's always sleep predominating over our lives. But sleep has something different as well from these other activities. For example, we can actually stop, we can willfully stop ourselves from doing any of these other tasks, including things that are essential for our life, like eating and drinking. People have gone on hunger strikes and starved themselves to death and so on. But sleep is something that's completely unavoidable. Even if we try to stay awake, something eventually kicks in, the sleep pressure kicks in, and we can't stay awake. So, for example, even if we're doing something enjoyable, like a sleep researcher studying a novel about sleep, that will still encroach if sleep pressure is high enough. And I've taken this example on the right here from Randy Gardner, who was a high school student in the 60s, and he tried to do as a high school project to stay awake. In fact, he's considered to be one of the most well-documented long-term sleepers. He stayed awake for 11 days. But the main point here is that after just a few days, it required 24-hour vigilance of his friends, basically distracting him to stay awake. And you look at his droopy little eyes there. It doesn't look like he's doing so well in being able to maintain his vigilance. So this sleep pressure eventually becomes something that's unavoidable. And this is something that drives, for me, to one of the major questions in the sleep field, which is, what is the nature of this sleep pressure, and where in the brain is it measured? It stands to reason that there's a place in the brain that is detecting how long we've been awake and saying, okay, you've been awake too long now, it's time to go to sleep. So in my lab, we take advantage of the observation of the last really 20 years that sleep is a nearly universal phenomenon. It goes back all the way to jellyfish, as demonstrated in this really nice paper from Nathal at Caltech in the upside down jellyfish, in which they showed that even the jellyfish, they don't even have a brain, they just have a neural net, but even they have what we can describe as a sleep-like state. And of course, since you're here on a international internet zebrafish meeting, fish is the one that we're going to take advantage of. And in zebrafish, the sleep research goes back all the way to this very nice paper that inspired me from 2001 from Irina Genova and colleagues. And then David Prober and I elaborated on this a bit in 2006 in zebrafish larvae. And then Yokogawa and colleagues in the Mignot lab extended this even further to adult zebrafish in 2007. So I say that sleep is nearly universal, but how do we actually know that? In mammals and birds and humans, we use EEG recordings, scalp electrodes, in order to detect changes in brain waves that are signatures of sleep states. But in many of these organisms, non-vertebrates, and even fish, this is a very difficult, if not impossible thing to do. And so what we use are behavioral criteria in order to define sleep. And so in brief, we define sleep using a whole range of behavioral criteria. The first being that sleep is an ultimately unavoidable period of inactivity. It's associated with a species-specific posture and location. You might say, for example, this sleeping lady from Malta in 3000 BC is exhibiting a very characteristic human sleep posture. Another really important element, though, is that sleep is associated with an elevated arousal threshold to sensory stimulation. This means that when you're asleep, you're less sensitive to your environment. Both visual, acoustic, and other kinds of stimuli take either more repeated presentations or stronger presentations in order for you to be able to sense those. And then the other two characteristics are that it's associated, it usually occurs with a 24-hour circadian rhythm. So it's usually confined to certain times of the day, or if you're not, or night, if you're nocturnal or diurnal, for example. And then there's also this compensatory sleep rebound following sleep deprivation. This is what we've already briefly touched upon. If you stay awake longer than you should and you deprive yourself of sleep, afterwards, the amount of sleep and the depth, the strength of sleep increases. So we use these criteria in zebrafish to define sleep states. And I'll walk you through some of the examples of the evidence for these features of sleep. So first, how do we track the fish? Well, we've been using for a long time this tracking software from Viewpoint Life Sciences, using a lot of our own bespoke analysis of that data that comes off. But this is what it looks like. We put the larval fish in a 96-well plate, and then we simply track when they're moving and when they're not moving. We're interested in these periods when they're not moving, and to define those as sleep states. And basically, you can see here that we can track 96 fish at a time, and with multiple cameras, we can easily scale this up to track, in my own lab, for example, more than 1,000 larvae simultaneously. And what comes out of this, if we look at a single larval trace, you can see here on the upper left that they're active during the day. They move around a lot, and then they become inactive during the night period. This is tracking them over several days and nights. But there's a lot of structure here. If we zoom in on this gray bar here, for example, on the right, you can see that the fish are active for maybe 10 minutes or so after the lights go off, or in this case, 5 minutes, I guess. And then they enter these long runs of these inactive bouts where they stop moving for minutes at a time. Then they have little wake-ups, tossing and turnings, I guess, and then they have these another run of these inactive bouts. And we measure the behavioral dynamics here by looking at the timing and the duration of these active bouts and inactive bouts. And by looking at how these inactive bouts correlate with the other features of sleep that I told you about, we defined all the way back in 2006 that sleep in larval zebrafish it is characterized by being inactive for at least 1 minute. So if the fish are inactive for at least 1 minute, they'll have changes in arousal threshold. So for example, if we now give them a stimulus here in the middle of this period where they're inactive, we can see that they're less sensitive to that stimulus. Here's one that we showed in 2006 just as an example, in which we're using transitions between light and dark stimuli. And you can see that the timing, this would be like a light alarm clock or something, if you want to think about it like that. And the timing from which the fish respond, if they were awake in the 1 minute epoch prior to the stimulus, they respond very quickly. But it takes 3 times longer if they had been inactive for at least a minute or up to 5 minutes in this graph, for example. So they have an increased arousal threshold. And this has now been shown by David Prober and our lab and others, that many other kinds of stimuli, including acoustic stimuli, also lead to this change in arousal threshold when they're in these inactive states. Now, I also mentioned that they're sensitive to the circadian clock and homeostatic sleep pressure. So the circadian clock is really this internal timekeeper that is timing the behavior of sleep to the rising and the setting of the sun. And this is the mechanism that goes wrong, for example, when you undergo jet lag and you now shift several time zones from one another. And the sleep homeostat is what is measuring how long, is measuring prior wakefulness and then driving increased sleep pressure as a function of that prior wakefulness. And this can be measured usually using sleep deprivation. In rodents, for instance, doing gentle handling, where you just kind of gently keep the animal entertained or aroused so that they stay awake longer. In humans, you can usually just ask them to stay awake longer as volunteers. And these two processes combine to sculpt the timing and duration of sleep. And it turns out we know a lot about the circadian clock, but we know much less about this sleep deprivation pathway. So how do we measure this in the fish? In the circadian clock, you can isolate the fish from external cues, like dark cycles and temperature cycles, and you can see that they'll nevertheless have a 24-hour rhythm. So here I'm plotting the total activity of the fish they were on a light-dark cycle for 5 days, and then we just turn the lights off and let them do what we call free running in constant conditions. And you can see that the fish nevertheless have an internal timer that says be awake when the daylight would have been and be inactive when the nighttime would have been. And you can see that it's not exactly, it's drifting slightly away and will drift over time, but it's just a little bit over 24 hours. Now, doing sleep deprivation to measure homeostasis in the fish has been much greater challenge, I should say. There's been many different attempts to do this, some more successful than others, I would say. And they all have some problems. For example, one of the very earliest ones was to use mechanical deprivation, some kind of shaking. You basically put the fish on a shaker, but this has some problems. One of the problems is that this causes physical damage. In our hands at least, we found that if you gently shake them at a very low level, they basically rock themselves to sleep, and so they can avoid or ignore the stimulus after a while, and so they don't really get deprived. And then if you do too much shaking, they're fragile, and so you damage the larvae. And so we found this was really not a very suitable way to manipulate sleep pressure in the fish. Gentle handling has been tried a couple of times. I'll show you an example in a second where David Prober and I, when we were postdocs in Alex Shear's lab, used a paintbrush method to just, we ourselves just sat there all night and touched the fish with the paintbrush every time they would go to sleep. And the problem with that is it's quite low throughput. So for the kinds of genetic and drug and other kinds of neuronal imaging studies that we might want to do, this was not going to really cut it. Some labs have used constant light, and constant light works pretty well. The fish find light as a very strong arousing stimulus, and so they will stay awake in response to constant light. But the problem is that light induces these circadian and masking effects. And so you can now have these confounds between the interaction between the circadian clock and the homeostatic process. And then finally, there's experiments that have used optogenetic manipulation, for example, drive the activity of wake-promoting neurons to inhibit sleep. And that's a pretty cool method. I actually think this is a pretty great way to deprive fish larvae of sleep. And this has been done in flies as well, for example, and in C. elegans. I would add the caveat that the light does interfere with behaviors. When you're using blue light, for instance, to manipulate channel rhodopsin, the fish do sense that blue light and will respond to it. So this works pretty well, but it also has its own problems inherent. So let me just show you the sleep deprivation gentle handling method. This was what David and I did when we were postdocs. We stayed up in the middle of the night, and we did deprivation for 6 hours here in the middle of the night, and then really sleep deprived ourselves. And then at the end, what you see is that we did get an increase in sleep in the deprived ones, but it only lasted for maybe an hour, and then they went back to baseline. So not only was this a heroic and difficult experiment, we didn't get very much bang for our buck, as you will say. So we've been thinking about this problem for a while, and a postdoc in my lab, Sabine Reichert, started to break it down into first principles, I guess. And she just started with some, very simple hypotheses. The first is that sleep pressure is a consequence of neuronal activity. There's already some evidence for this in a variety of studies in humans, rodents, etc. And so this wasn't exactly a big leap, but by thinking about this, allowed her to make real progress on this question. So she rationalized that if that's true, then we should be able to activate neurons indiscriminately, perhaps even, and that should drive acute sleep pressure changes. If you drive a lot of neuronal activity, you should drive a lot of increase in sleep pressure, and maybe you can accelerate the buildup of sleep pressure and drive increases in rebound sleep. And so once she had that idea, she came up with a very simple but powerful assay. And that assay was to wash on wake-promoting drugs, and then the drug is quickly taken up in the larvae and can penetrate into the brain at these early ages. We're talking between 5 and 10 days post-fertilization. And then wash the drug away, and then measure how much rebound sleep you get afterwards. And you can also measure how much brain activity you get in response to the drug. And so that was the simple assay that she came up with. And based on some old literature that had shown that, for example, patients that receive electroshock therapy have a very strong increase in sleep pressure. And also epilepsy patients, after grand mal seizures, for example, become incredibly hard to arouse. And so she decided to first start with a very strong wake-promoting stimulus, induce a seizure. And so she decided to induce a seizure in the fish and see what happens. There's a great amount of literature using the GABA receptor antagonist PTZ in larvae. You can see I picked one nice example from Mai et al. In 2013, in which they added PTZ and tracked the larvae. You can see that afterwards, the amount of activity that you get behaviorally dramatically goes up, and this behavior is very unstructured. So it's not like they're just running around. They actually have very disorganized behavior here. And you can see also that this amount of activity is dose dependent. So you increase the drug and you get many more seizures. Now, you can also see that using immediate early genes like CFOS to measure neuronal activity. Here's an example from our own lab in which you can see the control brains basically are blank you don't get, I mean, you have to stain them for a really long time to get neuronal activity under baseline conditions with CFOS marker. But after PTZ treatment, you get massive upregulation all throughout the brain, large amounts of CFOS being generated. And other labs have shown, like the Baraband lab, for example, have shown that these are bona fide seizures, both electrophysiologically and behaviourally. Okay, so the idea was just to wash on, give the fish a seizure for an hour, and then wash the drug off and see what happens. And to my, I should say, to Sabine's perhaps not surprised, because she really conceived this experiment well, but to my surprise, this worked. We wash the drug on here during this window here. You get a dramatic reduction in sleep and increase in activity. Then you wash the drug off. They immediately stop having seizures, but they enter this very long period of inactivity where they basically are asleep for 90% of the time. Compare that in the yellow, which is the treated fish, to the blue trace, in which basically they received vehicle control. Now, nicely, this is not damaging the fish in any long-term way, because the next day their behavior returns to normal, and as far as we can tell, it doesn't induce anything like apoptosis. We're not damaging the brain or anything like that. So that's with a very strong stimulus. But what about something that's a little bit gentler, or at least a different kind of mechanism? So we started taking our shelf of drugs and started adding them, these wake-promoting drugs that we knew worked in the fish for a really long time, and we started going through those. And I'll show you caffeine, for example, also did this very nicely at doses that don't induce seizures in the fish. So they're higher doses than what Will talked about in the previous lecture, but they're still not inducing anything like a seizure. Very high doses of caffeine can. And anyway, what you can see here is that this dose dependently increases the amount of rebound sleep that you get after this drug. And then also that night they return back to normal, and the next day they're normal. And you can see here on the right that this is a very dose-dependent response to caffeine. So again, we started grabbing things off the shelf. And one of the drugs that we had identified many years ago in a drug screen so just to conclude here, that just to emphasize what's different about this experiment, is that rebound sleep can be decoupled from the duration of prior wakefulness. They're acutely driven to wakefulness for an hour, and then afterwards you're getting this massive increase in rebound sleep. This is much stronger amount of sleep than you would expect from just gentle handling, as I showed you before, for example. So we started grabbing more drugs off the shelf. One of the drugs that I had discovered in 2010 is this drug aconitine, sodium channel modulator. And you can see here that compared to these baseline traces of 7 other drugs that don't really do very much, this aconitine induces a massive amount of wakefulness and physical activity. They're not having seizures. If I showed you a movie of these fish, they would look like just normal fish swimming around, but they just don't stop. They just swim and swim and swim and swim. So we were curious, like, okay, this was the biggest drug in terms of driving wakefulness that I had found in this drug screen. And we tested 6,000 drugs. So what would happen if we add aconitine and then wash it off? Well, to our surprise here, nothing happened. So here I'm plotting, I'm sorry it's cut off, but here I'm plotting on the left the cumulative sleep after the washout of the drug. So you can see that nothing happens. They're all on top of each other, basically. And you can see that here on the right. This dose dependence increase in sleep is completely unaffected in aconitine. And so that was a big surprise to us. What's going on? But it did have actually one implication, first of all. The first implication is that sleep rebound is not related to total physical activity. It wasn't that we drove the fish to be like super active and then their muscles got tired and then they went to sleep because they were exhausted. these aconitine fish have massive amounts of physical activity, and yet they don't show any rebound sleep at all. So what is it related to? Our first clue came when Sabine did CFOS expression following the exposure to these drugs. And what she found was that, as we expect, PTZ, caffeine, other seizure-inducing drugs induce massive amounts of neuronal activity, but aconitine, curiously, does not. It does induce some activity in the telencephalon here and in the ventral, in the posterior hypothalamus, but much less activity than we were getting in response to caffeine. So we hypothesized that either the aconitine does not engage critical sleep pressure-inducing neurons, or somehow the brain is able to sense how much neuronal activity is going on in their brain and then driving changes in rebound sleep. So yeah, so you can see here that, in fact, we can predict how much sleep rebound there's going to be just by measuring globally using RT-qPCR for the amount of CFOS expression that's induced within the entire brain. You can see that we can very strongly predict, it very strongly correlates with the amount of sleep rebound that you're going to induce. I'm showing this graph for caffeine, but of course this is true for all of the drugs. And this aconitine also gave us a very nice control to allow us to do some RNA sequencing time courses to identify sleep deprivation-induced genes. So the idea was we wash the drug on and off for an hour, we let the fish enter this rebound sleep phase, and then we measure how much sleep they got at 4 hours, and then we measure RNA, we do RNA sequencing here to look at gene expression changes. And we required 3 things to be true. First was that during the drug phase, that the gene expression would be low, and then the gene should come on or be induced during this rebound phase. But importantly, it should be unchanged in this aconitine drug. If it's really a sleep rebound regulating gene, it shouldn't be activated in aconitine because the aconitine fish don't have any rebound. And this is nice because this allows us to control for the exposure to very strong wake-promoting drugs. And we get a list of many genes that were increased in this case, but one of the very top ones immediately drew our attention for reasons that will be clear in a minute, and that is the gene Galanin. Galanin is an inhibitory neuropeptide. It's expressed in the hypothalamus and in the preoptic area. You can't really see the preoptic area is out of focus here, but you'll see it in a few slides. And Galanin has been implicated in sleep in rodents before. So this immediately drew our attention. And what you can see is that during this no rebound control, there's some Galanin expression. You can see this here in the hypothalamus. But after inducing strong neuronal activity for an hour and then washing it off, measuring 4 hours later, you see that you get massive increases in Galanin. We can detect many more Galanin positive neurons, in fact, and they're stronger. So Galanin comes on very strongly during this rebound phase. And in fact, Galanin tracks very nicely with drugs that can induce rebound sleep. So here's the control. Here's drug like the seizure inducer PTZ. You can see it induces massive amounts of Galanin. Same for caffeine. But aconitine, importantly, is no different from control. It does not induce Galanin in aconitine, as you would expect if Galanin might be important for regulating sleep homeostasis. Just to show you some quantifications of this, the amount of Galanin transcription that we see as measured by qPCR correlates very strongly with the induction of CFOS globally. And if we know how much Galanin is produced, we can predict fairly accurately how much rebound sleep we're going to get as you change the dose of the drug. So it seems that Galanin correlates strongly with both neuronal activity and the subsequent induction of rebound sleep. Now, neuropeptides like galanin very often increase their transcription in response to the activation of these neurons. As when neurons release peptides like galanin from their synaptic cleft, they have no reuptake mechanisms, unlike neurotransmitters. They can't really reuptake the galanin, and so they have homeostatic mechanisms in order to induce more transcription and translation of these neuropeptides. And so we thought that this might be possible, that this induction of galanin is related to the activation of galanin neurons during this rebound sleep phase. And so to look at that, so the question is, does this gene expression reflect changes in neuronal activity itself of the galinergic neurons? So to look at this, and there was reason to believe that this might be true. People have hypothesized for a long time in rodents that galanin neurons of the ventrolateral preoptic area were sleep promoting and mutually inhibitory with wake promoting neurons, such as the locus coeruleus and hypocretinergic neurons. And the idea would be that these are driving sleep and simultaneously inhibiting waking. So we were excited about these neurons. And so Sabine did this whole brain mapping method for neuronal activity. This is a method that was based on a paper from Owen Randlett when he was in the Engert Shear lab at Harvard. They called it mapping, and it uses essentially the ratio of phosphorylated ERK to the total ERK as an immediate early activation, basically as a measure of neuronal activity. Phosphorylated ERK very strongly correlates with how much activity that you have in the neuron. And so you can make these maps where you take whole brain imaging of antibody labeling for these two markers, and then you morph those into a reference brain atlas, and then you can map where the neurons are relatively up or down regulated in rebound sleep brains versus control brains. And what you can see is, I'm only showing this for the PTZ, but it's also true for caffeine, that we get very clear induction of an increase in neuronal activity that correlates with the location of these galanin neurons. This is a single optical slice through the ventral part of the fish brain, and we're looking down from the top. So here in the preoptic area, we also see here the galanin in the hypothalamus for reference, and you see some activation there as well. So it appears that these neurons are not only increasing their amount of galanin transcription, but they're also active. Okay. So is galanin essential for rebound sleep or not? In order to do that, we used CRISPR-Cas9 mediated gene editing to make a galanin knockout. And we have two different knockouts, but basically we get an early truncation, early stop codon and truncation, where the mutated galanin has essentially no mature galanin peptide. We can see that on the right here by Western blot. Here is the galanin, it's an antibody, by immunohistochemistry. This is an antibody staining for galanin. You can see there the preoptic and the hypothalamus. But in the Galanin mutants, we basically can detect no immunopositive staining, suggesting that this is a true bona fide knockout of galanin. And so what happens? Well, first of all, they're viable and fertile and seem pretty normal. If you measure their baseline sleep over 24-hour light-dark cycles, we do see a reduction in sleep. Here in the left, you can see here's a trace of sleep over time. And you can see in the purple trace, the mutants do have a little bit less sleep than either the homozygous or heterozygous animals. And this reduction is about a 10% reduction in total sleep. That's actually pretty modest. We expected perhaps this to be much stronger. However, what about sleep rebound? We actually see now that it is essential for this induction of sleep rebound after drug exposure. So for example, if we now wash on this caffeine, and then wash it off, we can see that here's the increase in rebound sleep from in wild type animals, but in the mutant animals we see basically no increase in rebound sleep. So they are unable to induce sleep after this exposure to drug. You can see this on this cumulative plot even more clearly. Here's the control. They induce a lot of sleep rebound, and here's the mutants, essentially just as much sleep as in a wild type control. Okay, so you might be thinking at this stage that, okay, fine, Galanin is required for this drug-induced effect, but it's drugs, and how is that really related to the normal kinds of sleep homeostasis, which really is measured by using physical sleep deprivation, for example. And so we decided that was an important question, and we revisited some of these gentle handling techniques in sleep deprivation, and Sabine came up with one which we call the optical treadmill. This is essentially you put the fish on a static grating here on the left, for example, and as a control, and then you expose the sleep depriving group to moving gratings that go left and right, and the fish will do their optomotor response and move to the left and the right following this grating. And they find this incredibly salient, which means that they will do this forever. Well, we don't know forever. They will do this at least all night, okay? And so we then compare this forced treadmill sleep deprivation technique to the controls and ask what happens. Now, if you do this all night long and then measure their sleep the next day, you can see here that in the wild type animals that are sleep deprived, they have an increase in sleep relative to the controls. But the Galanin mutants utterly fail to increase rebound sleep in response to this physical deprivation, suggesting that it's not just this drug-induced phenomenon, but it in fact is generalized to sleep pressure induced by gentle handling. So just to, the other question that might be burning in some people's mind, maybe not in this community because this is a zebrafish conference, but one question is Galanin actually required for sleep rebound in mammals? I mean, this might be just a fishy thing. In mice, this might not be relevant at all, or in humans, is this really relevant? Well, excitingly and kind of coincidentally, within a few weeks of publishing our paper, Bill Widsden and Nick Frank's lab in this nice paper in Current Biology, Ma et al., demonstrated that if you ablate the Galanin neurons of the lateral preoptic area, at baseline here and then do gentle handling, you eliminate the effect of sleep deprivation. So for example, on the left here, this is a measure called total delta power. This is essentially a measure of the coherence of slow waves that you see during sleep, measuring with EEG now, not with behavioral measures. And you can see that after you sleep deprive a control group, they increase this delta power. But the ones where you ablated these Galanin neurons in the in the preoptic area, you see no increase in this delta power. Similarly, the accumulative amount of sleep usually goes up after, this is the increase in sleep rebound after sleep deprivation. But the ablated, the mice that have these neurons ablated do not show this increase. So just to summarize this part of my talk so far, I showed you that this drug-induced rebound sleep correlates with the global neuronal activity, and that in turn leads to increases in Galanin expression during this rebound sleep phase and the activation of these Galanin neurons. And finally, by showing mutant analysis, we showed that Galanin is required for both physical and drug-induced sleep rebound. And so I think the overall implication of this is that sleep pressure is influenced not just by the duration of how long you've been awake, but by the intensity of waking experience. And I think many people would anecdotally say that tracks well with their own experience. And there have, in fact, been some experiments in humans, for example, giving people an enriched environment with lots of novel stimuli to think about versus being in kind of a boring room. And they've seen that they get increases in delta power, i.e., there's more sleep pressure in those people who had the exciting day versus the boring day. Okay, so just checking my time here. It looks like I have enough time to do the next part, so I'm going to spend just a few minutes going through this method that I wanted to tell you about, some new larval zebrafish behavioral analysis methods. And this was just published in eNeuro, and this is the work of PhD student Marcus Ghosh. And if I go too quickly or I'm too confusing in this stage, or my internet goes out, please do go read this paper. I think it's pretty exciting stuff. So Marcus wanted to bridge timescales from short behaviors to these long behaviors. What I've been showing you so far is in the first half of the talk, we're talking about sleep behaviors. Those are minutes long periods of inactivity and measured over hours and days, like here on the right. But in fact, the fish, when you measure them, if you look at their kind of acute bout structure, they have bouts that are on the order of sub-seconds. So I take, for example, here on the left, this work from the Orger lab in this beautiful paper in 2018, in which they skeletonized the fish and they tracked the tail beats here at very fine resolution. You can see here they're using a high-speed camera, and you're measuring this over hundreds of milliseconds, very quick behaviors, and you can see this very beautiful repeated bout structure of activity and inactivity that happens very fast. And Marcus was frustrated by the fact that essentially we throw out a ton of data here. We're like, well, we only care about this long-term stuff, and so we're not going to really look at this sub-second behavior. And he thought that we might be missing a lot. But there's a problem. The problem is that with this high-speed camera detection, the amount of data that's generated is huge. And that means that several things happen. First, you can only measure this for like maybe a few minutes. You can't measure this over several days, continuously in the same animal. And then also it means that they tend to ignore these kind of inactive bouts, and they only focus on the little parts when the fish are actually moving. And of course, we're interested in the parts where the fish aren't moving. So Marcus wanted to develop some methodologies to bridge these short and these long time scales. And so to do that, Marcus started with basically the same kind of analysis that we use to measure sleep, but took it down to a much finer detail. So I showed you before, when we track the fish, we detect when the fish are moving or not moving using this Viewpoint Life Sciences software and hardware. And what we get is basically pixel changes per frame for a fish. So in the given fish above, you see the fish is here, and then the fish moves with a little right turn here. And you can see that we detect some pixel changes during that movement. And so Marcus basically just took all these different features of these little bouts, the maximum, mean length of this bout, and called these active bout parameters, and then measured also the amount of time spent on a frame by frame basis, the amount of time spent in between these bouts. This could be a burst and glide movement, this could be a sleep bout. And then he took that data from 100 and some fish and 30 million active and 30 million inactive bouts, and he used unsupervised clustering to identify structure within that data. And he found that you can break these behaviors up into 5 active, based on these measures, 5 active and 5 inactive bout types, which we called modules. Now, just to highlight them on the left, we're showing here now the 5 modules organized from the least number of pixels to the largest number of pixels that change during a single bout. But of course, keep in mind that this is organized by including all of the parameters. And you can see that there's, basically different types of bouts here that we can detect that have different durations, pixel intensities, and so on. Similarly, although this is only one-dimensional data, we also found 5 inactive modules with basically 5 subdivisions of inactive modules, ranging down from very sub-seconds, basically, all the way up to tens of minutes. So from these active and inactive modules, we can now ask, how do these active and inactive modules change over time? How are they used over time? And just to highlight this, I've now plotted, well, Marcus plotted all the fish in the rows here, and then this is over day-night cycle, 2 days and 2 nights. And what you can see is that as we would expect, the active modules predominate during this day phase and are less predominant during this night phase. And conversely, the inactive modules go from being very short, so they have very short inactive bouts during the day, and then they switch to these long, bluer inactive bouts during the night, where this is basically the longest bouts and the shortest bouts. So you can see that they have this behavioral switch in their module usage. Another way to plot that is to look at the mean frequency across time. So you can see that the different bouts, these are now showing the mean frequencies of the different bouts or modules. And you can see that, for example, module 5 is predominant during the day and has a very high frequency at these transition periods when it goes from light to dark. And in general, you see that the active modules are more active during the day and the inactive modules are more active at night. But inspection of this shows surprising results that we didn't anticipate or know about. So, for example, this inactive module 4, these are the not the longest, but the second longest inactive bouts. You can see that they have this very interesting structure where their frequency increases as the day progresses, here like this, and they peak right at the transition from the lights on to lights off, but they also have a very interesting transition when the lights go from off to on, which was unexpected. So by analyzing the module usage, we can uncover interesting things about how modules are used and are chosen differently across 24 hours. So from those modules, we can look for higher order structure, which we call motifs. And Marcus used this dictionary-based compression method that was adopted from some work that was done in C. elegans to look at postures. And the way this basically works is you take each of the frames. Here, this is just a sample of a fish, a mock fish with the different pixel changes across time. You convert those data frames into modules like this, and then you give those modules a sequence. So this would be module number 1, then module number 2, number 1, number 2, number 1, etc. And then from those sequences, you look for rules. You look for basically runs of sequences that recur using some various rules that I don't have time to tell you about. But basically, for example, here you find in this sequence that they have a 2-1-2-1 pattern, and then they repeat another 2-1-2-1 pattern, and so on. You can see these repeated 2-1-2-1 patterns. And what you do is you reduce that to a new motif, which you call 4, and now 4 is a stand-in for this pattern of 2, 1, 2, 1. And so now you have a new sequence, which is 1, 4, 4, 3, 1, 4, 4, 3, 1. And then you look for patterns within that sequence. And you see now here you have a pattern of 1, 4, 4, 3, 1, 4, 4, 3. You reduce that to a motif 5. That stands in for 1, 4, 4, 3. And now you basically can compress this sequence from this long frame-by-frame data down to a shorter sequence of repeated patterns. And then you can ask, how frequently do they use these different motifs? And is there structure to those motif usage? I would ask you if that made sense, but we have to go on because that will be confusing if you guys all rang in. So if you look at this, we identify across the data set 46,000 motifs. These can last up to 20 modules long, meaning that the fish repeatedly uses the same sequence of 20 modules over and over again. And that can be on the order of 11 minutes long, considering that some of these can be long inactive bouts. The median number of sequences is 8. So most of these motifs are at least 8, or on average are 8 units long in general. And so we can identify these different kind of sequences that are used repeatedly by the fish. And now we can ask, are they used in different ways? Also, here's some examples just to make this a little bit more concrete. So you can see here, for example, this motif 4 is a little short bout followed by a long inactive bout, and then a more active bout like this. And you can see that we can get different burst and glide structures. There's an active bout, inactive bout, active bout, inactive bout that repeats. So we can get these different kinds of example motifs that are being used in the fish toolkit over and over and over again. So what can we do with these motifs? Well, one of the things we can do is we can ask, are these motifs chosen in different behavioral contexts? For example, environmental conditions. So we can find motifs that are selectively enriched, used very often, or constrained, used not very often during the day or the night. And we can then take those enrichment constraint scores and use that to segregate the behaviors. And you can see here in this T-SNE plot that we can reliably distinguish the day behaviors from the night behaviors just by motif analysis. And it takes only 10 to 20 motifs in order to do that. It's a segregation. This is also true. We can find specific day-night transition motifs. For example, they have a very characteristic startle motif that is very frequent at the transitions, both lights on to off and off to on. These were motifs that would not have been apparent in either the module structure or the analysis of just the delta pixels. So this is telling us something about the behavioral repertoire and how it changes across long time scales. We can also use this motif analysis to find motifs that are selective for different drug conditions. Let's go back to the seizure-inducing drug PTZ. I told you earlier that PTZ induces a very disorganized behavior, and in fact, this motif analysis can find those disorganized behaviors. And not only can it find conditions that are enriched in the control, these are very highly constrained behaviors in the PTZ fish. Basically, the fish can't do that normal bout behavior any longer. And then what was amazing to me, I have to say, is that by using these motif enrichment constraint scores, we not only can tell drug versus no drug, we can also distinguish between different doses of drug. In other words, as we increase the dose of PTZ, we're inducing different types of module and motif structure on the fish. This is even true for more subtle drugs like melatonin, which induce sleep in the zebrafish. We can actually tell what dose of melatonin the fish received just by analyzing these motifs. Okay, I'm aware that I need to wrap it up now, but I just wanted to say that these motifs are really bona fide structure, and we have a variety of ways of looking at that, but one of those is to ask what's the classification error? If we use just these motifs that are enriched and constrained in the different conditions, what's the error rate at which we make a wrong call for a given fish? And you can see that if we use just the modules, we have lots of errors in the calls. We can not, for example, tell the difference between some of these melatonin doses using just the selection of modules. But if we now use the motifs to make classifications, we can make almost perfect predictions. In fact, with PTZ, it is perfect with very few motifs. And so we can use basically this motif and module analysis to not only look at how behavior evolves over time, not just 24 hours, but also developmentally. We can also use this to ask what happens when we look at mutants or drugs, and how is the motif and behavioral structure being altered in subtle ways that you would not detect using just the normal methods that we've used in the past. So just to summarize this, the behavior can be organized into these modules and motifs. This organization allows you to bridge sub-second behaviors up to day-long behaviors, and that the motifs can be used to classify drugs, mutants, environmental manipulations. Okay, with that, I need to finish. I'd like to thank my lab and the people in the lab and my funders. I want to highlight again that the work in the first half was done by Sabine Reichert, a former postdoc in the lab, and the last half was done by a PhD student, Marcus Ghosh. I would like to thank the organizers for inviting me. This has been interesting so far, and I'm looking forward to the rest of it. Thank you.